The first question we ask is how does multiple sclerosis develop? And in reality, it's, there we go. The question is can we look at genetics? Do we look at the pathology of multiple sclerosis, which tells us about immune cells going into the brain? I like to think if we actually look at response to therapy, that will tell us more about multiple sclerosis than many things. But unfortunately, people have looked at animal models to tell us how multiple sclerosis works. And they've told us that multiple sclerosis is a T-cell mediated, CD4 T-cell mediated disease. And that's really shaped the therapeutic ideas. And so we think in terms of therapy that MS is controlled by T cells, which is a type of immune cell. And if we look at the genes, there are 200 that we've discovered so far. The main one is called the HLA-DR, and that tells us that there's CD4 cells are important. So again, it's given us the idea that CD4 cells are important in disease. And the pathologists kind of have even convinced themselves that CD4 cells are important. But when you look down at a microscope, in fact, it's actually the CD8 cells that are much more common. And I'll tell you that the HLA-DR can be involved in actually viruses infecting cells. And what I'm going to try and convince you that actually MS is not a T-cell disease, it's a B-cell disease. And if we look in the animal models, they don't really respond to B-cell therapies because MS is a uniquely human disease. So here are the MS drugs. There's a lot of them. So there's beta interferons, glutathione acetate, dimethyl fumarate, daclizumab that is thought to increase natural killer cells, but that's been kind of withdrawn. Then there's teriflunomide that works by being anti-proliferative. It stops T cells growing. And then there's people think there's a depleting drugs, so alemtuzumab, ocrelizumab, and cladribine. They deplete T and B cells. And then there are drugs that stop migration. So fingolimod that traps T cells, keeps them in the lymph glands, so they can't get into the blood, so they can't get into the brain. And natalizumab that stops white blood cells getting into the brain. So it's quite complicated. But I want to make it even more simple for you. So one way to view it is actually most of these drugs are actually anti-proliferative. So the beta interferons, because they're potentially antiviral, they will block the viral machinery that is involved in proliferation, and it will stop T cells proliferating. A dimethyl fumarate targets a molecule called GPR109 and can stop T cells proliferating. And daclizumab blocks interleukin-2, which is involved in T-cell proliferation. But that's still complicated, so I actually want to make it much, much simpler for you and to say, actually, they all work the same way. And this is probably heresy, and this is what I want to try and convince you of some new kind of research that we've been doing to suggest that actually the drugs all work the same way, which makes it easier to think how you can use them. Then you just have to think about the side effects and things like that. Now, part of the problem about thinking how do MS drugs work is because the T-cell immunologists, which tell us that there are different types of T cells, there are CD4, there are CD8s, there are memory, there are naive cells, very complicated. But if you look at the B cells, they just say, well, there's one type of B cell, the CD19 positive B cell. And until you actually realize that they aren't just a single population, you can't understand how the drugs work. Now, I said response to therapy is important. And if you look at T-cell therapies, they really haven't worked very well in multiple sclerosis. So CD4 T-cell depletion, depletion of cytokines that make Th17s don't really work. So how does the B cell be important? So what you need to do is to actually just remember there are a number of different types of B cells. So B cells are made in the bone marrow, and then they become pre-B cells and the immature B cells. which mature, they circulate in the blood, and then they go into the lymph glands. And there they see their target, and then they grow in places called germinal centers. And then they can either make memory cells, which remember what they've seen, or they can become plasma cells, and plasma cells are the ones that make antibodies. And what you need to remember is that cells come from the bone marrow, and the memory cells come from the lymphoid glands. And the memory cells can take years to generate. So this is important. So if we go to how does alemtuzumab work, this is what we see. So on the left panel, you can see in blue that there's a massive depletion of the CD19 B cells, and by 6 months, they're back to the normal level. And if you look at the T cells, which are on the right-hand side, you can see that they're depleted, and they're gone for years. So we know alemtuzumab lasts for years, and so that gives you the idea, well, B cells are back to normal very quickly. It's got to be the T cells. But the important thing is to remember that that CD19 population is different types of cells. And one of the cells are the immature cells. And so what happens with alemtuzumab is it doesn't appear to purge the bone marrow very well. And we can see that in transgenic mice. If you look at the yellow bit, is the bone marrow. And if you give alemtuzumab to mice, it can deplete the T and B cells, but it doesn't really purge the bone marrow very well, which means that the B cells in the bone marrow can rush out from the blood, which is what you can see on the right. You could go up 200% their normal level. And then they mature, and you can see that they're 120%. And what's happening is they're repopulating in the absence of the regulatory cell. And this is one of the reasons why alemtuzumab can cause autoimmunities that occur in about 50% of people. But what this is masking is if we look for the memory B cell, we can see that the memory B cell, like the T cell, is depleted for many years after alemtuzumab. And what we did is we looked through all the MS drugs, and we found that every single one of them depleted memory B cells except natalizumab, which increased them, but their job is to stop migration into the brain, so the effect is still a relative decrease. And there was a thing called atacicept, which caused an increase, and that made MS worse. So every drug that works in multiple sclerosis depletes these memory B cells. Now let's have another look at some real life. So if we concentrate on the top corner, we can see beta interferon, which we know isn't a highly active drug. If we ask the question, if we look at, say, 20% depletion is a relative mark, we can see that beta interferon works well in a small population of people, which we know is what happens for multiple sclerosis, whereas mitoxantrone, which we know is a more effective drug, you can see actually depletes memory B cells in the vast majority of people. So if you look on the right, we can see glutarium acetate and beta interferon. You can see there's a big spread in the level of depletion. So the drugs that don't work very well don't deplete the memory B cells as well, and the ones that work very well, like fingolimod, deplete the memory B cells well. So now we have a hierarchy. If we look at natalizumab, you can see on the left. If we look at the T cell level on the top, you can see there's no difference. But if you look at the bottom of the memory B cell population, you can see that their levels are actually increased in the blood because they're trapped there. They can't get into the brain. But of course, if you stop using natalizumab and don't switch people onto treatments quick enough, you get rebound. And it's because perhaps the memory B cells are all lined up in the blood waiting to go. Now, a new drug that's on the cards is ocrelizumab. That's done so it depletes memory B cells for the long term. And what you do is you get a treatment every 6 months. So if you look at the CD19 population, you can see that they're back to normal within 6 months, which would say, well, let's treat every 6 months. But remember, it's because of the immature cells coming out of the bone marrow why it looks like it's going back to normal, and the memory B cells, as you look, are depleted for years. And in some other cases, it may be 5 years that these are depleted for, suggesting that these drugs can actually have a longer effect than from treating every 6 months. And indeed, when we actually found some trial data from ocrelizumab, you can see on the top, you were treated with either a placebo or in blue beta interferon or in yellow ocrelizumab. After 48 weeks, so we were switched onto ocrelizumab and it worked very well. And then people got nothing for 18 months on the far right. And you can see it was still working. So you might not have to treat every 6 months, but it's telling us that there's a commonality of how these drugs work. So alemtuzumab is what we call an induction therapy, so you only have to give it for a short treatment, and then it lasts for a long time. So the drug is active. So how do we tie this up to biology? And what we know of multiple sclerosis is there's a genetic component, but we also know that virtually everybody with MS is infected with EBV. Now, EBV infects B cells via CD21 and HLA-DR. This is the MS gene. And what it does is it makes memory B cells. So the virus that we think is triggering MS is making memory B cells, and we can see that in transplants. You can see in the top with somebody who didn't have EBV, and in bottom you can see the red blobs. That's because they had EBV, and that's because it drives memory B cells. And we have CD8 cells which are accumulating in the central nervous system, and some of them are there because they are targeting Epstein-Barr virus. So it all links in. So memory B cells can present antigen to T cells, so maybe this is how it's working. But what EBV does, it makes things called latent membrane protein 1. And these molecules kind of mimics to stimulation by T cells and by the B cell antigen receptor, which means that the B cells become independent of T cells. And so it's possible that EBV is the driver here. B cells are the key driver in MS, and it may be that you don't really need T cells that much. So in conclusion, B cells, we believe, are important targets for MS, and they can drive a demyelination because they produce toxic factors for oligodendrocytes, and they can also create niches in the brain whereby the B cells survive for a long time and they produce antibodies, and these may stimulate cells such as the microglial cells, which can then probably drive the progressive disease when you have demyelination. So in conclusion, I think B cells are important targets for therapy in multiple sclerosis, and if we think about that, it gives us a unifying mechanism of what maybe causes multiple sclerosis, the pathology of multiple sclerosis, and importantly, its response to therapy. So I realize there's a lot to take in there, but potentially it means actually MS treatments got simpler, not more difficult, because if you view them as all working in a similar way, you don't have to worry about, how does this drug work? They all work by targeting these cells. Then all you have to worry about is side effects and other things. So thanks for listening.